respectable Dean Faculty of Sciences, Professor Dr. Sher Mohammed, distinguished scholars, esteemed researchers, respected colleagues, students, ladies and gentlemen, Assalamu alaikum. Department of Biology was established in 1999. Currently, we have 3 BS programs, including BS Biochemistry, BS Botany, and BS Microbiology. We recently have launched MPhil program in Microbiology and Molecular Genetics. We are trying our level best to uplift the department in the community of science, and this conference is part of this mission. On the behalf of Department of Biology, I extend a very warm welcome to all of you to this keynote session. We are truly honored to have 6 distinguished speakers with us who will be sharing their expertise and insights on the diverse aspects of biology. Among them, Dr. Benjamin and Dr. James Cox are joining us online to share their valuable thoughts. We are equally delighted to welcome our esteemed guest, Dr. Awais Rashid and Dr. Xiang Gao, who are present here in person from China, a country with which Pakistan shares a bond often described as higher than Himalayas, deeper than oceans, and stronger than steel. We also extend our warm welcome to Professor Dr. Shahid Mahmood Beg and Dr. Khalid Khwaja, whose valuable talks will further enrich this session. Now I request Dr. Sheng, Dr. Awais, Dr. Khalid Naeem, Dr. Shahid to please come on the stage. Ladies and gentlemen, let me begin this keynote session. It is my immense pleasure to introduce our first keynote speaker, Professor Dr. Shahid Mahmood Beg, honored with Hilal-e-Imtiaz and Sitara-e-Imtiaz. He is an eminent scientist and a distinguished academic leader. Currently, he serves as Dean Faculty of Life Sciences at Health Services Academy, a professor at Aga Khan University, Karachi, and adjunct professor at CAM, Lahore. Professor Beg has held prestigious national and international positions, including advisor to OIC Comstech, Chairman Pakistan Science Foundation, Director General National Institute of Electronics, and Head of Health Biotechnology, NIBG. His lifelong contributions in molecular genetics and science policy making continue to shape Pakistan's scientific landscape. He has authored 161 research articles in high-impact journals. He has supervised 39 PhD theses in the field of human genetics. Today, he will share his insight on state of recessive diseases in Pakistan, challenges and opportunities, a topic that is highly relevant to public health, highlighting challenges and opportunities to manage rare genetic disorders in our population. Please give a big hand to Professor Dr. Shahid Beg. Please, sir.
Currently, thank you, Dr. Rizwana, for the very extended introduction. I'm at Health Services Academy as a Dean of Life Sciences. And before that, I was Chairman of Pakistan Science Foundation and professor at Aga Khan University. And so I have been trying to do science, to promote science, and to inspire our younger generation to continue in science. So that's why I happily accepted your invitation, and I'll try to share my experience and with our young students and researchers, what are the challenges and what are the opportunities of doing science, and especially in the biological sciences and in genetics. You see, we are a very big population, more than 225 million people. We are the 6th most populous country, and moreover, we have the highest population growth rate, one of the highest population growth rate in the world. We are increasing with the rate of 2% every year. So if we continue at this stage, in 2050, we will become the 5th largest population. We are growing at a faster rate than India, than Bangladesh, and Sri Lanka, etc. Pakistani population is a heterogeneous, it's a mixture of populations and multiple ethnic groups. And in all these groups, the practice of cousin marriages is very high, consanguineous marriages. It is around 70%. So 70% of the marriages in Pakistan are cousin marriages. Either these are in brotherly or tribe or in the same family. So this practice, it adds to the incidence of genetic diseases. If you look at this map, you see the dark blue areas. These indicate the highest rate of consanguineous marriages. And Pakistan is here. You see, we are practicing the highest number of cousin marriages in Pakistan, followed by other Muslim countries. And if you look at the rate of cousin marriages in the Muslim countries, you see overall Muslims are practicing 50% of the marriages which are cousin marriages. In other religions, in most of the religions, it is not allowed. In Christianity, in Hinduism, in Sikhs, in Chinese, etc. In some countries, it is prohibited by law, but in Muslim countries, it is allowed. And if you look at the rate of percentage, Pakistan is the champion in cousin marriages. 70% of the cousin marriages are consanguineous marriages, followed by Saudi Arabia. You see, it is 67%. But the problem with Pakistan is that we are 225 million people, 25 crore log, whereas the population of Saudi Arabia is only 3.5 million people, which is 35 million, which is 3.5 crore. And Saudi Arabia is a resource-rich country. They have a well-placed health system and healthcare system, primary healthcare system, whereas in Pakistan, even in some of the areas, we don't have the primary healthcare system, and we are a resource-limited country. So what is the solution to this? The solution is genomics, and genomics is a multidisciplinary science. So it includes biochemistry, biology, medical sciences, and bioinformatics, and environmental sciences, epidemiology, microbiology. So it is a multidisciplinary science. And when you talk about genomics, it is always on the low priority at the policy-making level in Pakistan, because they look at the more prevalent and more visible disease burden, which is of infectious diseases. They look at COVID, influenza, dengue, malaria, hepatitis, TB, etc. So these, they call them rare diseases or orphan diseases. But I tell you, in Pakistan, these are not rare diseases. Me and my group, we are working from the last more than 3 decades on these diseases. And the number of affected individuals is increasing every year. And these are no more rare diseases in Pakistan. So that's why this is a big challenge. And by collaborating and making partnerships and inviting our fellow scientists to work together to accept this challenge and continue working on this to find some solution for these diseases. So most of the work which we carried out, it was during my active service at National Institute for Biotechnology and Genetic Engineering. It is located in Faisalabad. And these are the type of diseases, the pedigrees, which we identify from the field. These include intellectual disability, deafness, eye disorders, skeletal disorders, neuromuscular disorders. There are more than 8,000 inherited diseases which have been classified so far, identified so far at clinical level, but not at molecular level. At molecular level, only half of the diseases have been characterized. So to treat any disease, whether there is any treatment, or to prevent and to diagnose, it is the first thing to work on the molecular genetics of these diseases. And identifying the disease and the disease variants, it's very challenging work. It takes many years, needs a lot of money, resources, team, and infrastructure, and high-end equipment to analyze these diseases and to find out the genes. So this knowledge will provide the basis for the treatment and the prevention of these diseases. Currently, most of the genetic diseases are incurable. There is no treatment. Only some of the diseases are curable if these are diagnosed. So we identified more than 3,500 large families who were practicing cousin marriages, and they have 2 or more than 2 affected children. This is the strategy to identify the disease gene. You see, first is to travel to the remote areas, because most of these families, they don't visit the hospitals. If they ever visit the hospitals, they are told that there is no treatment for these diseases, so you better stay at home and दुआ करें और बस अल्लाह पे छोड़ दें वगैरह वगैरह. So we go to the remote areas, identified these families, and first take their consent. This is a research study, and you will be diagnosed, and we will provide them the genetic counseling and ways to prevent the disease. And if it is diagnosed, and if there is treatment or there is any prenatal diagnosis, we will let you know. So we start with the linkage analysis using the microsatellite markers, and if it is linked, we go for the mutation detection by using the Sanger sequencing. And now we are using the gene panel, whole exome sequencing, and Mendeliome. Mendeliome is the set of all the known human disease genes which have been identified so far. And ultimately, we go for the whole genome sequencing. Then statistical analysis, calculation of the large score, and if it is a novel gene, in many of the cases, we find the novel gene which is responsible for causing this disease. And when you identify the novel gene, it means you have actually reached the cause of the disease. And when you find out the novel locus, you find the disease gene and the genetic variant which is actually causing the disease. So when you identify the genetic variant, this is the drug target for the precision medicine. So if it is curable, these families can get treatment. Although the treatment of these diseases is very, very expensive at this stage. But when it will become popular, people will know about the genetics and the genomics and the treatment of the diseases and application of NGS and other technologies in this part of the world. Then the cost of these diagnoses and the treatment will gradually become lower and lower, and in the affordable capacity of our local population. So this is how we traveled to the different areas of Pakistan, in Punjab, in Sindh, in Balochistan, and Kashmir, and in KP, using various mode of transportations. And you see the genetic counseling session with the families to tell them what is the disease, tell them about the basics of the genetics, about the cousin marriages, and how to live with the disease, and what are the options, if there are any, for the treatment, etc. Now, if you look at the evolution of the human brain, this is the most fascinating organ part of the human body of any animal. And if you look at the brain sizes of our close relatives, that is the chimpanzees, baboons, gorilla, and macaque, etc., you see the human brain is the largest one. And this is which differentiate from the other primate animals that we are human. And this part, which is known as the cerebral cortex, this is the most important part. This part which is involved in the cognition, perception, memory, and languages. And this part makes us different from the animals. This is the difference. And you see, these are the patients of primary microcephaly. ये जिन्हें हम शादोले कहते हैं ना, छोटे सर वाले. They have a small head circumference and small size of the cerebral cortex. Small cerebral cortex. This is the patient MRI, and this is the control MRI. So this is the difference, and this is the important part of the human brain. That's why we are different from the human. So when we study these patients with the microcephaly with small head circumference. At one stage, on one hand, we are trying to understand the disease mechanism. We are identifying the disease genes which are causing the disease and the drug targets for the future treatment. And at the same time, we are understanding the human evolution, the evolution of the human brain, why we are human and why the monkeys are monkeys, baboons are baboons, and the gorillas are gorilla, why they can't speak, why our cognition is better than other primates, and why we can communicate in any language, in Urdu, English, Chinese, or whatever we speak. So this is the most important part of the brain. So by studying these families, at one hand, we are providing the genetic basis and the future treatment for these diseases, incurable diseases right now, but in the future, inshallah, these will be curable. And on the other hand, we are understanding the mechanism which is involved in the development of the human brain. These are the type of pedigrees which we encounter in the field. You see, this is one family, and there are so many affected with primary microcephaly. And the people, they are living in remote areas, and they don't know this is an inherited disease. They have different taboos. They think it is some badua from or something like that. They have several misconceptions about this disease. And this is what we are doing. We are approaching them and providing them the counseling and taking the sample for genetic analysis. And in this cohort of these families, we have 700 patients, 860 in total, 700 from Pakistan, and 241 from primary microcephaly, and some other from other neurodevelopmental disorders, the disorders which are mainly involved with the brain development. It leads to various level of intellectual disability and brain development. This is the current strategy which we are using now. If there are students of molecular biology, genetics, and biology, they should be more interested in this type of analysis. The strategy, what we are doing here, based on our prior knowledge, since we are working on these diseases from the last many years, now we know where are the founder mutations of these diseases. For example, in case of microcephaly, we know there is a very common gene, ASPM, in the Pakistani population, and we can first screen the founder mutation by a very, very simple test of PCR, restriction site mutation assay. And 50% of the families are resolved. And then we go for gene panel analysis. We were also using the Mendelium. Mendelium is the assay for all the known genes, 450 known genes which are known so far, human functions. We screen these. Then if it is unresolved, we go for whole exome sequencing, then the homozygosity mapping, and if it is still unresolved, then we go for molecular karyotyping or copy number variation analysis. So this is the strategy, and in case of skeletal defects, since we are working on various diseases, we have various approaches to approach the molecular diagnosis. So after the analysis, if you look at the our results, out of 241 primary microcephaly families, we have found the mutations in 102 families in the ASPM gene, which is the most common in Pakistan. Then 69 in other known genes. And during this study, we found 29 novel genes. You see, my colleagues who are sitting here, who have the knowledge in the molecular genetics, they know how important and significant it is when you find a novel gene for any human disease. So during the analysis of these 241 microcephaly families, we found 29 novel genes. If you see from the universal perspective, this is a very big contribution to the medical science. At one end, as I said, we are understanding the human brain development mechanisms, what are the genes which are involved, and at the same time, we are diagnosing the causes of the genetic causes of the disease, and these are the future targets for the precision medicine, for the gene therapy, for the gene editing, and treatment of these very challenging neurodevelopmental disorders. So 4 gene causing mutations were found in the modifier regions. This is another discovery that disease causing genes can be in the modifier regions as well. And after using all these methods, 81 of the family still remains unresolved. So we are working on these, and hopefully we will be able to resolve in the next few months or few years. So these are the novel genes which we found during our analysis. You see, and when you find a novel gene, especially which is involved in the human brain development, then it is accepted in the highest impact factor journal. You see, some of our one of our paper was published in American Journal of Human Genetics, then Nature Genetics, Journal of Clinical Investigations, etc. So these are the genes, and these are the phenotypes. These are microcephaly and intellectual disability and developmental delay, etc. This was the first family which we analyzed during our genetic analysis. You see, again, there are so many affected, the black boxes and the circles and the square. These represent the affected individuals. You see, these are the forefathers are same. They continued cousin marriages, and the result is the homozygosity of the disease genes, which leads to affected children. And currently, I should again mention that there is no treatment for these neurodevelopmental disorders. So these are some of our results which were published during the PhD theses of our students in various journals. And one of the figure from our publication was able to find the cover page in the medical genetics in one of the volume, one of the issue. This is our very interesting study on the microcephaly and the neurodevelopment disorder. This is actually the primordial dwarfism, which causes the primordial dwarfism, and the microcornea as well, or the cataract. So you see, this is again a big pedigree, and these are the patients of this family. And for this, I should also share what is the role of collaborations and partnerships and research networking. You see, when we work in isolation, one thing is very clear: you cannot work in isolation in any science, especially in the life sciences. It is a multidisciplinary science. You need a lot of money. It is not a easy science. You need high-end infrastructure, equipment, and teamwork, and not only one specialty. It is a multidisciplinary science. So for this modeling of this disease, we collaborated in Edinburgh with Professor Andrew Jackson. He has a state-of-the-art zebrafish facility for the to study the disease gene model. We inducted him in our team, and he provided us the zebra gene modeling and the function analysis, etc. So we were able to publish the PLK4 in Nature Genetics by this collaboration, and there are 35 authors in this paper, and there are 7 countries which are involved. So what PLK4, which is the protein which is involved in the centriole and the cilia. You see, if the PLK4 activity is normal, we have normal centriole, normal number of centriole and the cilia. And if it is when the activity decreases, in the case of heterozygotes, the number of centriole and the number of cilia decreases. And when it is an homozygous affected patient, then there are very few centriole and very few cilia. So it leads to dwarfism and microcornea or a neurodevelopmental disorder. You see, when you work in the collaborations, research networking, then you are able to do good science. And this was one of the purpose of my talk today, to emphasize on the importance of collaborations and networking in science. You see, if you don't do any collaboration, you cannot do genomics or biological science in Pakistan sitting in silos or in your own lab without sharing your data, without sharing your expertise, your opening your door for your colleagues. So if you establish such a collaboration at national and international level, only then your work is accepted in the good journals. So publishing in good journals, it means the other scientists will read your science, and you will be contributing in good science. And you are contributing for the mankind, for the disease treatment, solutions for the issues. And this issue is a very big issue, not only in Pakistan, but all around the world. You see, human health is the most important thing. हमसे सबसे ज़्यादा पैसे अपनी हेल्थ पे लगाते हैं, अपनी शक्ल पे, कपड़ों पे, देखने में. So this is the most important thing. These are the genes and the functions of the genes which we have elucidated and reported that these are the genes and these are the functions where these genes are involved. So these help us in understanding the disease mechanism and the function of these genes in the human biology and genetics. Again, the intellectual disability in Pakistan, because of the large family size and the cousin marriages, the intellectual disability incidence, if you look at the global incidence, it is 1 to 3%. In Pakistan, it is reported as high as 6.5% for mild. Intellectual disability ranges from mild to severe. It can be mild, it can be moderate, it can be severe. And there are estimated 5 million patients with intellectual disability. And for autosomal recessive intellectual disability, more than 570 genes have been known so far. So it is very heterogeneous. The diagnosis and the treatment is a very big challenge in this area. So the skin disorders, how we work and report, this is hypertrichosis, sparse hair, genetic, and the nail dysplasia. And in one of our study, by studying analyzing a Pakistani pedigree which has a nail dysplasia, we were able to find the gene, the Frizzle6 gene, which is involved in the development of the human nail. So when you studying these families, you identify which genes are involved in the making or development of certain organs, for example, nails or brain or anything in the human body. Until now, I have been showing you the recessive diseases where the parents are carrier and they are likely to have, there is a risk of having affected children. But these are recessive disorders where parents don't have disease. Now this limb deformity, polydactyly or syndactyly, where people have more or less fingers or digits in the upper or lower limbs, it is a autosomal dominant disease. You can call it a disease or a trait. It is autosomal dominant. Here, one of the parent is affected. He has more or less fingers, and he or she will have affected children, and they don't care much about it. They think that it is only extra digit or a less digit, and it doesn't make any difference. It is just a cosmetic problem. If you have an extra finger, you can cut it down surgically, and you can have 5 fingers. And if you have less finger, you can have a happy life with 4 fingers or 3 fingers. There is no issue. But in some of the families, there are marriages between 2 affected individuals. They are cousins, and even if they are don't cousins, they are not cousins. If both are affected, both have less or more, then they are likely to have very severe phenotype in their children. You see, when there is a marriage between 2 affected individuals, then in case of autosomal dominant disease, the phenotype is very severe. They don't have any finger, they are not able to walk, they are not able to hold anything. So it is very important to provide them the genetic counseling. And in Pakistan, we don't have any genetic counselors. If you go to the West, in any hospital, wherever you go, whether it's a primary care, secondary care, or tertiary care, they do have a genetic counselor. Even if it is a cancer hospital or any hospital, they have a genetic counselor who tells you about the risk of having this disease in other healthy individuals of the family. And what are the options if the disease is there? Are there any treatment options? Is there any early disease diagnosis system available in the country? So in Pakistan, on one hand, we are trying to create the disease awareness and provision of cost-effective premarital screening and genetic counseling, and finally, the disease gene discovery and treatment option and other strategies for the control of these diseases. So these are other diseases. I should finish as well. I will skip some of the slides which are not very important. This is one of the family with profound deafness who can't hear and who can't speak. But in this family, there were sudden deaths as well. Deafness doesn't lead to sudden death. When we extensively studied this family, we found that the affected children, they are dying because of the brachydactyly. Brachydactyly is a slow heart rate. So the gene which is CACNA 1D, which is involved in the deafness, it also leads to slow heart rate, which leads to the death of the affected individual. So we worked extensively, and in this case, we involved the biophysicist, biomathematicians, biostatistician, clinician, and it was a very big team. We worked for almost 2 to 3 years on this family, and ultimately found that this is the mutation, this is the gene, which is, which is causing the slow heart rate and death of the affected individuals. So now this family is having a changed lifestyle, and from the last 10 years, there is no more sudden death in the affected individual. So this paper was declared one of the best paper of 2011. It was published in Nature Neuroscience, and it was awarded with the Gears Foundation Award in Germany. These are some prenatal diagnoses in the recurrent pregnancy loss. You see in Pakistan, you see there are multiple pregnancies, and in some cases there are recurrent pregnancy loss. So we worked on this, and we found out in diagnosis in many cases by using the whole exome sequencing, and prevented the affected births and provided them the prenatal diagnosis and premarital diagnosis. And this is a very challenging and very exciting family of brain tumor from Islamabad region. In this family, there were so many deaths because of the brain tumor, glioblastoma. And if you look at the pedigrees, you see there are very strong consanguinities. All marriages are cousin marriages. First, the double line indicate a first cousin marriage. You see there are so many deaths of glioblastoma. And you know, cancer is not a typical Mendelian disorders. It is not inherited in a autosomal recessive way in most of the cases. But there are 2% of the cancer which are inherited in autosomal recessive manner. So if you look at the pedigree, this is a typical autosomal recessive pedigree of glioblastoma. We worked on it extensively, and finally we found the mutation in the PMS2 gene, which is the gene that is involved in the DNA mismatch repair mechanism. So there, only because of the deletion of a T, it leads to DNA mismatch repair, and it leads to glioblastoma. Now this family is having regular prenatal diagnosis. We perform various prenatal diagnoses of brain glioblastoma here in Pakistan, and ultimately we were able to diagnose a healthy baby in this family, and the family was overjoyed, and they shared their sentiments with us through an email. These are other examples of cancers. So what we are doing by using the next generation sequencing to identify the disease genes, so they can have the counseling and the premarital and the genetic counseling and early disease risk assessment and carrier detection, etc. You see, in Pakistan, the most common genetic disease is the beta thalassemia. It is a transfusion-dependent disease, and the affected children, they require regular blood transfusion. The allele frequency is more than 6%. It means there are 12 million carriers of thalassemia. Each year, we are having more than 10,000 new affected children with thalassemia, and currently there are more than 100,000 registered cases of thalassemia who are on regular blood transfusion. What we are doing, first we establish a simple arms PCR method for the detection of mutations which are common in Pakistan. We provided them the cascade testing, the trials testing, and extended pedigree analysis, etc. Welcome, Professor Ramzan. Thank you. I am at the end of my talk. You see, we identified this family by this index child, and this family, you see, it's a big pedigree, and they didn't know that this family is suffering from thalassemia major. Most of their children, they die undiagnosed. So when we diagnose this child with thalassemia, and we extended the cascade testing to the whole pedigree, you see there are so many carriers. The half-filled circles and squares, they represent the carriers. So these are the families which need the carrier screening and genetic counseling to control the disease at least expense. These are the result of our prenatal diagnosis. We provided the prenatal diagnosis, that is a diagnosis in the third trimester of the pregnancy by taking the chorionic villus sample from the pregnant woman. And if you look at the prenatal diagnosis results, it is typical Mendelian ratio. 50% are heterozygous, are carrier for this disease, 25% are normal, and 25% are affected. Affected means they will require regular blood transfusion throughout their life. So these families, they opted for termination of the pregnancy because they cannot afford, it is very expensive, iron chelation therapy is very expensive, and it takes a lot of money and time and psychosocial problems for the family. So most of these families who already have a sick child, if they have another sick child diagnosed during the pregnancy, they opt for termination of the pregnancy, and it is allowed. In Pakistan, we have a fatwa from Mufti Taqi Usmani way back in the 90s that if any fetus which is suffering from an incurable disease, then the termination of pregnancy is allowed in the 120 days of the pregnancy. So this is what we are doing from the last 3 decades. Now, how to prevent these diseases? You see, some of the diseases, as these are being characterized, some of these are preventable, some of these are curable. Most of these are preventable because the treatment is very, very expensive. If you talk about gene therapy or precision medicine for any rare disease, it is in millions of dollars. And no one can afford in Pakistan. So the way to control these diseases is by prevention, genetic counseling, premarital screening, and screening in the school, etc. In case of thalassemia, we have identified the mutations which are most common. In case of primary microcephaly, we also know that 77% of our population, they have ESPM1 gene, so the diagnosis is very simple. As I said, restriction site mutation assay we perform, and the most common mutation is screened, and 77% of the cases are resolved by this simple PCR method. The way forward in Pakistan to introduce and popularize the genomics in the hospitals and in the clinics and research laboratories. Genetic counseling facility, you see, we have to focus on the prevention of these diseases. The treatment, as I mentioned, it is very, very expensive. Even it is not affordable in the rich countries as well. Genetic counselors, I already mentioned, it tells about the disease and the outcomes and the options and how to live with the disease and how to avoid the disease, etc. You see, these are the collaborators. This work would have not been possible to lead to any success or any logical end without the collaborations. And I have been lucky and blessed to have wonderful collaborators all around the world. And they are very humble, जिस तरह के बस हम हैं उसी तरह के वो हैं. Very good scientists, very humble, very cooperative, very generous. These are in Germany, in USA, in Switzerland, UK, USA, France, China, Germany, Denmark, Germany, Sweden, Germany. So these are very generous, and they are happy to establish the research partnership, collaborations, and networking. And we have been working with them from the last more than 2 decades, and we have been able to do some good science only with this collaborations, networking, and partnerships, and sharing of resources and expertise. And this is what is all about integrative biology. You see, you cannot understand any biological mechanism if you don't involve multiple expertise. And the future of genomics and the translation research in Pakistan, if we look at the size of the Pakistani population and the disease burden in Pakistan, we must have a center for genomics in Pakistan. I have been trying from the last more than 10 years, although I have been sitting as the helm of the affairs as a chairman of Science Foundation, but it is very hard to convince our authorities. I hope one day they will be understand and realize the gravity of the burden of the disease. मैं तो ये कहता हूँ कि we are sitting on a time bomb. We are heading towards a genetic disaster. We have to control it. We have to avoid it. जब तक हम ये जीनोमिक्स को पॉपुलराइज नहीं करेंगे, लोगों में अवेयरनेस नहीं करेंगे, तो यही पीरी फकीरी और देसी नुस्खों से काम ये नहीं चलने वाला. We have to control it. This is the type of infrastructure which we need: laboratory infrastructure, trained personnel, computational infrastructure, educational resources, and the community engagement. This is very important. Unless the awareness is not there, genetic counseling is not there, genetic screening is not there, precision medicine is not there, we will not be able to control the ever-increasing disease burden in Pakistan. So this is the layout of this genomic center, which I have been proposing from the last more than 10 years. But inshallah, I'm very optimistic. जब इतना काम कर लिया तो ये भी हो जाएगा. This is my research group, which was in Faisalabad, way back in the health biotechnology. And this is my core group of human molecular genetics. This is a part of my group of human molecular genetics. Many of them were working in the labs of collaborators. Now they are almost all of them, they have completed their PhDs. They are working in Pakistan. Some of are in US, in Denmark, Sweden, Germany, and they are contributing, and they are very enthusiastic and productive scientists in the field of genomics and integrative biology. And here it's a very long list of collaborators and the funding organizations, the donors. You see, again, I must say genomics is a very challenging field, but we have to continue working on it. And for this, we need a lot of partnerships, networking, sharing of expertise, and a lot of funding as well. In my projects, the funding was mainly from Swedish Research Council, from Ludwig Foundation, Denmark, Center for Molecular Medicine, from Cologne University, Germany, and Higher Education, and Pakistan Science Foundation as well. At that time, I was not chairman when I received the funding. And this is the very first pedigree which I identified from the field in the south of Punjab. You see, these are all cousins. They are suffering from a skin ectodermal dysplasia. And you can see the hope and the expectation in their eyes. They are looking towards us.
Our next keynote speaker is Dr. Sheng Gao, who has joined us all the way from China, and we are truly honored to have him here in person today. Dr. Sheng Gao, holding a PhD in bioinformatics and plant breeding, is a member of Crop Intelligent Design Algorithm and Model Innovation Team at Institute of Crop Sciences, Chinese Academy of Agricultural Sciences. His research employs bioinformatics and machine learning approaches to investigate the evolutionary dynamics of crop genes at pangenome level and throughout plant development. In addition, he also investigates the regulatory mechanisms of crop genetic network. Dr. Gao has led several important research projects including Hainan Provincial National Science Foundation General Project and a subtask of a key scientific and technological mission from Chinese Academy of Agricultural Science. He has co-authored over 40 research articles published in esteemed journals such as Molecular Plant, Journal of Integrative Plant Biology, Plant Cells, a New Phytologist, Plant Journal, and the Theoretical and Applied Genetics. He also holds 3 national invention patents. So let us extend a warm welcome to Dr. Gao.
A great honor for me to be here for having me here at this very wonderful conference and meet a lot of distinguished professors, researchers, and meet with some junior scientists and very talented student here. And I'm Sheng Gao from the Institute of Crop Sciences of CAS, which is Chinese Academy of Agricultural Sciences. I want to briefly introduce about this. My institute is the largest and the top one crop science institute in China. And now I'm also belongs to the team of Nanfan Research Institute of CAS. That's a new department of our academy. So today my talk is about our teams recently working in applying AI for research in crop genetics and breeding. And according to the 2025 Global Report on Food Crisis, nowadays 673 million people worldwide are facing hunger. And I'm not sure why is always sorry about that. And the basic solution for for this challenge, for this crisis, is to breed new varieties with high yielding and high quality varieties. But for conventional breeding strategy, there are several challenges at the moment. One is that the trait of the crops normally is controlled by complicated genetic architecture, and also most of them are severely influenced by environment, and this reason make the genetic improvement cycle of those crops is very long and very low efficiency. So at this moment in the area of artificial intelligence and new biotechnology, the new technology-driven breeding strategy will be a very important support for germplasm innovation. And to answer this, to respond to the challenge, the crisis, our team proposed a triple drive strategy, which converging biotechnology, breeding big data, and artificial intelligence together. For biotechnology, we already have, like in the past 30, 40 years, marker-assisted selection and genomic selection. And nowadays we have genome editing and also some other new biotechnologies. And for the breeding data, in China we have accumulated a lot of genomics, transcriptomics, and even epigenomics data, and also the high throughput phenotyping data. And now we are also systematically collecting the environmental data from field. And for the artificial intelligence part, now machine learning, deep learning, and even recently large language model have been proposed to predict or generate some that result we desired. And in this framework, we want to integrate all this like BT, DT, and AI together to generate a new breeding strategy. And we believe that this strategy is the core engine of next generation of digital research. And the major question that our team propose is to develop the AI, how to develop the AI-based genomic prediction models using like huge amount of multiple omics data to achieve the multi-dimensional and accurate prediction of complex trait of crops. So this slide shows the roadmap of our lab, and we take the AI techniques as the central processors, and actually AI can now do very excellent in three aspects of our research. Sorry, one is the first aspect is for gene mining to develop the AI-enabled algorithm or models to discover the more genes and excellent alleles that we can use for breeding. And the second part is to predict the complex traits of crops. And the third aspect is to build like the breeding decision system. In our studies, we call it smart breeding platform. We will combine all the algorithms and method into this platform to support the final breeding decision. So In my following talk, I will basically introduce two sections of our team's work and basically about the gene mining and genomic prediction, and we'll also show some practical case that we use, how we use this algorithm or models. The first part is AI intelligent gene mining algorithms, and we developed actually this algorithm based on the central dogma and spread from the different layers of biology, like from the genome and epigenome, transcriptome, proteome, and metabolome. And the first study is that we developed a deep learning-based method to predict the gene function, and in this work, we actually take all the sequence of the genes, the amino acid sequence of the genes in a given species, and put them through one hot encoding to convert those genes into a matrix and use a convolutional neural network to predict the gene function with the gene ontology an annotation of these genes, and use this method. We found a series of ion transporter genes, and those genes are never been found in the model species and well-studied crops. And we validate their ion transport ability in the wet lab, and found and their ability are validated. And we found that this method can be resolve the threshold dependency issues in the conventional sequence of alignment, and enable more accurate prediction of gene function. And the second study is that we also developed large language models and based method for annotating transcript or interpret transcriptomic data. And here we also use the sequence data from a given species and also the expression profile, and taking this data into the language model that we trained, and the model will take all the input expression profile and the sequence as a sentence in natural language to find the representation with the background. And it can be used to explore the regulatory elements or pathway and other key genes with completely unknown functions. And the second work, we also combined machine learning and co-evolutionary analysis to construct the genetic network. In this work, we first collect more than 300 species genome, and then we focusing on a reference genome, the reference species genome, and that one is the one you are interested in, and we can get the evolutionary signal of each of the genes from the 3,000 genomes, and get the co-evolution matrix, and then analyze the signal from each of the evolutionary trajectory. The basic idea is that when given two genes in the genome, if they are functionally related, then their evolutionary trajectory, in a more simpler ways, the absence and presence of status within each of the species genome that we used. might be correlated. And in this way, we can connect the known genes with unknown genes. And we also apply the machine learning to predict these genes across different species. And the fourth study that we use machine learning, we developed a machine learning model for screening the metabolic biomarker. And in this work, we use proteome and metabolism data. And we combined the basic machine learning model like gradient boost, random forest, and the supporting vector machine with the newly developed CAN, which is a deep learning model and deep learning structure, and put them together to predict the biomarker that we are interested in, and also the genes correlated with the biomarker. And with this method and models developed, we here's a showcase that we focusing on mining the halophyte genes to improve the crop salt tolerance. The halophyte genes is from a halophytic species which can grow in the coastal area in China and also in the North America, which is highly salt tolerant. It's highly tolerant. And we used the aforementioned method. We found a series of genes, and we validate their function in the rice system and found some of the genes can improve the salt tolerance of rice, and also can somehow adjust the grain shape of rice. And most of these genes are still under validation, and we got some confidence from this work, means our algorithm can be used to manning the genes or alleles from less studied species and then to used for improved crop. The second part is about intelligent genome prediction algorithms. So the first method I want to introduce is deep neural network genomic prediction, which is also known as DNNGP. This model have a very simple architecture, has only 3 convolutional neural network layer, and followed with 1 batch normalized layer and 1 flattened layer and 1 fully connected layer. And the convolutional, the CNN layer is responsible for extracting the representation or patterns from the input data, let's say maybe SNP array, SNP data, and also transcriptomic data and other type of omic data. And the following layers were use these representations to predict the performance of a given genotype. And based on our experiment, this simple deep learning models outperforms the mainstream method by at least 10%. So it's simple, but better performance. So because of the simple architecture, it have a very good, it shows a very good computational efficiency. So DNGP can, take slightly longer, may take a slightly longer time than traditional method on smaller data set, which is normally less than 500 samples. And when considering thousands of samples, DNGP is at least 10 times faster than other algorithms. And now our method is released on our GitHub repository, and also the data. We also regularly update the software, and we also held seminar about this software. We also held the seminars and the workshops for this software regularly, and to help our collaborators to use it. And based on this knowledge, we also want to know how about when integrates environmental data would improve the genomic prediction accuracy. So as we know that the G by E influence crop traits severely, and in this paper we take maize as an example, and we found that when integrate the environmental data with the GBLOB, the classic GBLOB or RBLOB method, we found for both grain yield and grain moisture content, the prediction is improved. And also the most interesting thing is that with this framework, we can predict the trait performance across different environments when you include the environmental parameters of different places. And next method is called AutoGS, which is more, we go a step further to use this, we developed this method, and it can maximizing the genomic prediction performance by using the environmental data. But this method, it reduced the environmental parameters dimension first according to the growing period of different crops or plants. And in this way, it can increase the prediction accuracy by at least 14%, and the auto-tuning process can reduce the training time by more than 200 times, and still keep quite high accuracy. In addition, we also use the SHARP technique to enable the quantifying genetic and environmental contributions for researcher and breeder. Another method is EXGEP or XGEP. We developed this method also include for handling the environmental data, but for this method, there's no prior knowledge you need to put into this method. So this method can directly integrate the genotype, weather, soil, and other environmental parameters to generate a model that's suited for the given species, and it also shows very high performance. So now we combined the AutoGS and EXGEP into a package called a G by E toolkit, which is also can be found in our GitHub homepage. And I'm not sure whether there are plant breeders or students or researchers who are working in plant breeding. I hope the people who in the audience is interested with this method can use it, and we are very keen to know your comment and your feedback that can help us to optimize this software. And finally, is a application case for our genomic prediction model is we test all our method in the maize breeding design with integrating doubled haploid technique and genomic selection. So here is a classical maize breeding process, the first step is to select the parents from different heterozygous group, select the elite parents from different groups, and after that we can use the DH technology to generate two cross and use DH techniques and generate a lot of DH lines in very short time. And at this second stage, we can use the genomic prediction to select more elite DH lines as parents. And for the step 3, we make the cross between the elite DH lines and use a genomic prediction model again to evaluate the hybrid performance. And in this case, we generate about 2,400 DH lines, which is produced from 33 commercial hybrids, and genotyped them. And finally, after filtration, we got 437,000 high-quality SNPs. And with those SNPs, we can build the models and do population analysis. And we do selection sweep analysis between the DH populations and taking the 500 traditional inbred lines as a reference population. We found a total of 54,000 selection sites. And interestingly, 72.7 of former reported functional genes were found to be located within the selected region. And with those selected regions, we built the genomic prediction model, and we built the models for 8 yield and yield-related traits, including plant heads and the flowering times, etc. And for all of them, we acquired relatively high prediction accuracy, but the accuracy is varied because the different heritability of different traits. And based on these models, we finally select a small group of elite DH lines, and then in fields do crossing and we basically use test lines for 2 sowing area in China. One is early mature area, the other is a mid-late maturity area, and a total of 11 environments. The field data were collected, and here we use the genomic prediction model again with this data to train a large model to filter out all the possible crosses between the elite DH lines. And finally, we select like 2 excellent combination. Now is under the second year of regional province or regional trials, and they will probably get variety approval. And yeah, that's basically all my talk. Finally, I want to acknowledge our amazing AI for breeding team, which is led by Professor Hui Li, also from CAS, and also thanks to our amazing colleagues and breeding students, and also to all of our collaborators. Here is the QR code. If you want to find us, find more information about us, you can scan the QR code. And that's basically all my talk today. Thank you very much.
Thank you very much, Dr. Gao, for a very fascinating talk. Your insights into AI-driven technological innovation in breeding and intelligent genome prediction algorithms clearly highlights the power of bioinformatics and machine learning in advancing the agricultural science. We are grateful for your expertise you have shared with us.
I am delighted to introduce our next speaker, Dr. Khalid Naim Khwaja, a distinguished One Health expert with over 35 years of experience in research, teaching, and policy making. Dr. Naim played a pivotal role in controlling avian influenza in Pakistan by leading the National Avian Influenza Control Program. Later, he co-founded the first One Health Pakistan network in collaboration with Message University in New Zealand for zoonotic disease surveillance. A WHO-certified biosecurity auditor, he has supervised more than 60 postgraduate theses and published 70 peer-reviewed articles. Today, Dr. Khalid Naim Khwaja will share his expertise on strategy employed locally for substitution of antimicrobial-resistant antimicrobials with vaccine in combating antimicrobial resistance in food animals.
Thank you very much, the organizer, for inviting me for this topic. A little bit more about the background. Actually, I have been working at PARC for whole my career, and then later on I started a private entrepreneur where we were producing vaccines for animals. So the idea about this topic was just to discuss with the young researchers, like many of you are here, to see the fate of the research has to be a product at the end. That's what my perception is. So I would like to just share that how the recent issue of antimicrobial resistance in organisms is basically affecting the animal health issues from that perspective. Being a One Health person, I know it's kind of a joint effect of these antimicrobial resistance between the human health and animal health. So the focus will be basically to bring to your notice that there are a number of ways and means whereby we can handle this AMR stuff, and what we are doing it at an animal health side. That's the main idea of presentation. Being in the private sector, I'm involved in manufacturing vaccines for animals and pharmaceuticals for animal for the past now 10 years after my retirement from PRC. Thank you. Okay. I think the antimicrobial part is that in food animal, antibiotics are used obviously for raising the animal, and these antibiotics, as we know, by non-judicial usage, it creates resistance, and this drug resistance is passed on in the environment from the organism affecting the animal health to to human health, and this kind of interaction really makes this difficult nowadays, as we are seeing a lot of drug resistance happening in microbes involved in human health, and both human health and animal health as well. How it basically happens is that the AGPs, the antimicrobial growth promoters, are used in raising animals, some type of animals, primarily those which are fed in a closed feedlot system, where the antibiotics in a small dose becomes part of their growing procedure. And primarily these drugs are used at a very low level at very early stage of growth. Later on during production, they are withdrawn. But having said that, once it's in the system and the normal flora is affected and mutations and resistance developed, basically this contaminates the environment and passed on to the other organism in the environment, which eventually maybe end up in human or again in the animal production cycle. These AGPs basically are used in poultry in Pakistan because we do not have feedlot system of raising dairy cattle or sheep and goat. So our major problem in this country is in the poultry. And as I said in the early, that it is basically managed somehow because it's used at early stage of production, and they have very less effect to the contamination of the product like eggs or meat. However, while contaminating these organisms, contaminating the environment, they still have a chance to move into the environment and into the cycle of drug resistance in human as well. In other countries, AGPs are also used. In European countries, they are banned almost, but in many other countries which raise animals, they still use them. Now recently, for the past number of years, they are replacing these antibiotics of low doses to probiotics. And as you know, probiotics are the microorganisms which basically replaces the normal flora and help the animal to have a better growth and better production from minor GI tract infections. However, antibiotics are used used in human, and as we know that we don't have a organized dispensing of antibiotics in human sector as well, but we just to focus on the animal health. The treatment in animal health model is still more judicious because of the economic impact of this antibiotic usage. In human health, still we are saving one individual, but when you are treating in animal health model, a flock of maybe 100,000 birds, you cannot treat each bird individually. So it's very expensive to think about using a antibiotic for treating animals like chicken or any large scale, animal produced. So the bottom line is that we still have to find out better means, better way out how we can avoid these antibiotics, not only probiotics, but there are a number of other things so that may be discussing in my presentation. This is a basically pathway which describes how the antibiotics used in animal model passes through their different systems and contaminate the environment, and from the environment they again get back into the cycle of the human health, products like water or food products produced and other things, and eventually they end up that this AMR bacteria organism, they end up in the human health cycles. So what are the issues in Pakistan? I will be basically trying to address that, how we have managed over the years to overcome this particular component, not following the standard international approaches, because it's not because of some technical reason, primarily because of the cost and awareness and our limitations in our systems. But just to bring to your notice that what other activities are being undertaken to address this AMR. As we know, we have lack of data, and for usage of antibiotics in different sectors, we do not have a strong or organized surveillance monitoring system for the usage of antibiotics and the resistance developed in different organisms in the environment. And still we have lack of evidence, like is it originating by using these drugs non-judicially in animal health system, or it's from the human health system and going other way around. So a lot of discussion takes place that where do we have an organized system of antibiotic usage. And as you know, in our country, I think it's equally disturbing in both the sectors. And because it's very difficult to really specifically identify the major reasons behind that emergence. So by the time people are gathering more information, we are developing other systems whereby we can have better approach to address this AMR situation from human health perspective and animal health perspective. There are some other better way to substitute it, which we will be addressing. So what are the alternatives of antibiotics? As we know, it's a One Health component, as I said in the beginning, so it means it has to be weighed both way. Have to tackle it at the human health level and also at animal health level. So in case of the animal health level, there are a number of non-antibiotic alternatives listed down over here, and they are probiotics, there are some phytochemicals, there are certain immunoglobulin components, and some enzymes. They are also used as a replacement of antibiotics, and above all, there's a vaccine. So my focus will be about vaccine, that component further to bring you to your notice what we are doing in this case. Number of studies previously undertaken by other countries, they have different models where they have organized food animal farming system, where they have studies which has proven that use of vaccines in place of antibiotics has more beneficial effects and also more economical. Just for vaccine, from veterinary perspective, just to update your knowledge, there are primarily two types of vaccine which we use in animals. the live vaccine and killed vaccine. And then there are a number of further components of these vaccines. However, the main criteria is any vaccine which we identify has to be cost effective, it has to be very specific, it has to be effective in the sense of because there's a lot of mutation in organism, both viruses and bacteria. So you need to have a very precise vaccine developed specifically for certain organisms. In animal health side, there are multiple pathogens. Same is true for few human pathogens as well. But we know the influenza and salmonella are very common organisms, and we have many subtypes of influenza, and similarly number of types of salmonella. So to have an international vaccine, that's very difficult to really understand. So we have to develop our regional specific, serotype specific, homologous vaccine in different regions or countries. And that's where it brings us to a topic that if we have to use a vaccine, what quality of vaccine we can be using it. So main theme is that more homologous the vaccine strain is with the field strain, more effective it is. But the question is from where you can get such a vaccine. Antibiotics have a different way to use. But we have to understand that this can only be done if the organism is very stable in some cases, or we have to develop our own vaccine in different parts of the world. So, as I said earlier, there are a number of ways of producing those live or killed vaccines, number of simple ways, a number of new biotechnology-based methodologies available. We don't want to get into detail of that, but the whole thing is that vaccine has to control the disease, to minimize the losses, and because it's an economic game in animal health model, so we need to have a very specific vaccines which can either minimize the clinical signs when the animal is affected after vaccination, or it should not be affecting the production of the animal, like milk, meat, eggs, whatever. So these are the two targets for which we develop vaccines. And in some cases, like I just mentioned an example of a disease, rinderpest in cattle, some of the major approaches are to eliminate the pathogens from animal population. And one of the things happened in the rinderpest, it's a viral disease, and it was basically eradicated all over the world after a prolonged effort. Similarly, like polio in human. And the approach is in this the animal agriculture model that you eliminate those pathogens so that we can basically control the cost of raising that animal and also cost of producing the vaccine. So that's kind of a very, very, you can say, combined approach. So by definition, I think if we have a proper vaccination plan strategy, we can just help reduce the AMR development in animals. That's the theme. Okay. As I said earlier, that the animal vaccines have to be safe and efficacious. Still, some current animal vaccines have some issues, problems. That's why we keep on improving those vaccines by using the new biotechnology techniques. And that's why more research and more investments is required to basically involve in vaccine production business or activities in a country like Pakistan. When we see the effectiveness issues, another issue is the evolving of new pathogens. There are a number of organisms which are unique to our area, our region, and then that complexity of the organism's genetic makeup, its mutational capabilities, and also its complexity of pathogenesis, epidemiological part, all this information has to be available for developing a very specific vaccine based on our needs. So the main emphasis, my emphasis over here is that we cannot rely much on an imported vaccine which is not compatible with our needs. That's the whole theme over here. So to start with, what has been done in Pakistan was to prioritize the diseases and focus on certain diseases in which we can have a vaccine and avoid the antibiotics. So currently, it has been divided into two components. Like certain vaccines selected they fall in the livestock sector vaccine, and some vaccines fall into poultry disease vaccine. As I said in the beginning, that when you are going to treat your herd, your flock, you have to treat them holistically. So for 100,000 animal, you have to have medication. So it is a lot easier, lot convenient if you use a vaccine for all of them and provide the protection. And in case there's problem, infection, then you can individually handle some animals or have a sport medicine. So these are the listed different bacterial organisms which cause infection in animals, and vaccines are now developed in Pakistan in this regard. Another point to note is that viral vaccines are also used for animal disease control. I have not mentioned them specifically, but it's very obvious that when there's a viral infection, a secondary bacterial infection, it is affecting that animal. So if those secondary infection bacteria are those for which we already have vaccine, so that means those animal will perform better. So the whole idea of bacterial vaccine is not only to protect them from bacterial infections, but also to protect when there is a bacteria as a secondary infection. This is the structure animal population in the country. These animal species are just primarily focused on major dairy species. Poultry also, we are producing 1.4 billion chicks every year. That's a huge population. And you can very well imagine the types of medicine used, the drugs used for treating them. So it's a big industry. As far as the coverage of vaccine is concerned, because of the limited resources of the government, very selective livestock vaccination is used, because government is giving vaccine free of cost, but they cannot afford to provide all the animals. So the range is between 15 to 80% based on the type of disease. Some diseases, they have more vaccine production, some they have selective. But in commercial poultry, if you notice, 100% means they have to purchase it, and because economic game, so they are basically using 100% vaccine in poultry. So if we just emphasize on this one point, you can imagine how we can control the AMR in poultry, where we are using APGs and antibiotics, as explained earlier. What are our overall requirements of the vaccine, animal vaccine? So I've just listed down different species in which vaccine is required, but we are somehow not in a position to vaccinate all the animals. However, the major vaccines are used apart from the food animals. We are using this in pet animals, and there are a number of types of vaccine which are required to be done, and you can see the selection of our vaccines available in this country. So that basically indicates a gap not from business perspective, gap from the research perspective, gap from the more collaboration between institutions developing vaccine strain and those producing vaccines in commercial sector. So this is, I think, an opportunity to make if we focus on vaccine development in this country. What we are doing. So currently there are in public sector, there are 4 institutions in Pakistan at each provincial capital, and they are producing number of livestock and poultry vaccines. However, there's one unit in Sindh province, which is basically autonomous, and they're producing a number of poultry vaccines. In private sector, there are only 3 companies, in which one is in Islamabad. I also work for one of these one in Islamabad, and other companies in Lahore, they are in Lahore. So they are producing primarily a good number of livestock and poultry vaccines. So just to mention that what type of vaccines is available, and that will basically correlate with the positivity that by using these vaccines, we are replacing the use of antibiotics in the country. That's the whole theme of my presentation. So just take it from that perspective, that if we start using that part at this stage, maybe 5 years down the road, 10 years down the road, our AMR situation will be a lot better. And what I'm presenting here is basically WHO has now prescribed that at the end of the AMR, the best approach will be we revert to the vaccine development in human and as well as animals. But it depends, what your priorities are and how you can just take it up. Anyway, so this listed bacterial vaccines are selectively produced, prepared, and used in Pakistan. So I can say with confidence that with the presence of these major bacterial pathogens, we are saving a lot of antibiotic usage in this animal population. And this is not time to present all the data, but just give you a feeling that that approach of handling it from this perspective, how helpful it could be. From the poultry, the previous slide was about livestock diseases. This is the poultry and fisheries. And I think that's another wonderful to see that 5 different vaccines are used for bacterial treatment or control in Pakistan. And that has saved us, I think, a huge quantity of antibiotic usage. And this is a very unique example. And I will say we should feel proud of it, rather than all the time we feel criticized and thinking we are not doing anything or we are research as problem. All these conferences and gathering, I think, should come up with some achievements as well. That what we have contributed so far, and how can we contribute more if we develop our interactions, develop on this platform, or between the public and private sector, institutions. Similarly, fisheries, we are not using much vaccine in fisheries, because fisheries sector has just started few years ago. As we bring the animal to intensive farming, as we have done in the case of poultry, we will be switching towards that part. We'll be having more problem with the antibiotic resistance. We're using antibiotic non-judicially. We have to save them. It's economics game. It's nothing to be God-fearing or Allah se daro ji logon ki. No. It is economics. So it only can be handled through better economic replacements, opportunities. So you as a scientist have to provide replacement for something which you want other person not to do. That's what I simply believe. So this is a good indication that we have just started with the US developing first vaccine of fisheries in Pakistan. I'm sure a couple of months we'll be able to just get it registered, because this is the future, where future diseases will be coming, and we'll be, have no choice but to go back and start thinking about the vaccine. I think that was my, brief thesis, that we need to encourage our scientists, our institutions, to come up with the product. With all the science, whether you do it through biotechnology methods, you through some other standard, microbiological method. At the end, we as end user, now I'm standing on the other side of the wall as a kind of industry person, we need a product from the institution. Where is the product? So publication is good, student teaching is good, number of students good, but at the end of the day, we'll be asked, and I think number of people were here, Dr. Shahid and other, they've seen the new proposals coming to the forum of funding, and at the end of the proposal, because I'm a member of a number of committees like that still, and at the end of the day, there's no product at the end. So it's all imaginative science, I call it. I'm not saying it's wrong. I'm saying, please, let's also think about it, that there's a good potential. Just summarize what I said. Emergence is at the rise of AMR. AMR is now one of the biggest issues for One Health perspective, and we have to assess ourselves. Where are we staying? Which side of the wall? What pluses we have, and how we can improve based on our own conditions. The whole thing is. So I think apart from the some other approaches to contain that AMR, we can also think about selecting or developing the non-antibiotic alternatives. As I said, huge research is required for probiotic development, microbial standardized for, replacing, and also vaccines, as I said earlier. So the bacterial vaccines has to be prophylactic, as other vaccines are. But the good thing is that once you just use it, then you are basically avoiding for a number of months, or maybe not a year, to control that particular disease. And once you're in the track, I think that will be more helpful for managing the misuse of antibiotic. You can't stop it. It's going to be used somehow or the other. But if you stop the cause, I'm sure in the commercial side, nobody's going to give antibiotic for fun. Sorry. Another thing we discussed was that because of the intensive farming, as we are moving towards that, vaccine-based prophylactic approach has to be adopted for replacing the antibiotic. I also mentioned that in Pakistan, we are basically using the vaccines against bacterial and mycoplasma infections, and that's very successfully helping the farmers and the community to control those infections. If we could do more research, like if we could do the biotechnology part and produce vaccine in a more aggressive way rather than traditional ways, I think there is a huge scope of providing this vaccine more at economical prices to the farmer, so that everybody could use it. Just like polio vaccine, I think I don't want to give a discouraging remarks, but polio vaccine, we're getting a donation, and donation is going to stop in 2030, and then what we're going to do, and we're yet still thinking about it. So I'm somehow a member of that committee sitting there last month. The question was, what will we do? But we're not talking, can we produce it? So I'm here just to convey a message that please, we need a collaboration with the institutions who are doing research to develop the vaccine seeds, to develop new technologies, to offer to private sector, and let's develop our own vaccines. So the last request is that we have need a strong academic industry interaction, and that will be helpful for producing more effective vaccines. I think that's all. Thank you very much.
It's my honor to introduce our next keynote speaker, Dr. Pervaiz Rashid. Dr. Pervaiz Rashid is a senior scientist at CIMID based in China and associate professor at Quaid-e-Azam University, Islamabad. An expert in wheat molecular breeding, digital phenotyping, and genomics, Dr. Rashid has authored over 120 research papers and having more than 7,000 citations. His work on genotyping platforms and functional markers is being applied globally in over 40 institutes. Recipient of prestigious awards including International Collaboration Award, Dr. Rashid is a leading voice in advancing food security and sustainable agriculture worldwide. I am highly privileged to introduce Dr. Rashid today, not only as an eminent scientist, but also as my PhD supervisor. And he's always being an inspiration for me. He has always been guided me, and he has always mentored me to be a best scientist and excel in the field of wheat research. Please join me in giving a very warm welcome to Dr. Pervaiz Rashid.
Bismillahirrahmanirrahim. So first of all, I will congratulate the organizers of this conference. They did a wonderful job. And I am very privileged to be part of this conference that they invited me as a keynote speaker. So I will be talking about wheat genomics for the nutritional quality and health benefits. So if we talk about nutritional quality and health benefits, that is the complete framework that how we can impart these important phytochemicals and micronutrients by the wheat grain. And if you look at the phytochemicals and micronutrients, they are usually distributed in the bran and alluron layer. And that is a very good news for the Pakistani population because we usually consume chapati, which is made from the whole meal flour. And because we consume chapati, it means that we can get benefit from these phytochemicals and micronutrients as compared to the populations where they consume the whole meal flour, like in noodles in China. So Among these phytochemicals, one compound which is known as ferulic acid is very important. And it exists in conjugated form with the dietary fibers. It means that if we can increase the ferulic acid contents, we can also increase the dietary fibers. We can have a dual benefit. And it has demonstrated very good benefits, like it is a very important bioactive compound used in traditional medicine. It has anti-cancer effect, and it can regulate insulin sensitivity. And more important thing is that it has a very good stability during the grain storage and the processing. It means that this compound remains available after the product processing. So what we did in the beginning, that we tried to screen all the Pakistani wheat varieties for the ferulic acid contents, that if we have very good quantity of these ferulic acid contents or phenolic compounds in our wheat cultivars. And we did the field trials for 4 years in more than 200 wheat varieties. And the result which we came up, that in the modern wheat varieties, we have more phenolic compounds or ferulic acid as compared to older wheat varieties. Now this was a very striking result, because we know that micronutrients like iron and zinc, they are more in the older varieties. So, but these phenolic compounds, they are more in the modern varieties. So the only plausible explanation which we have, because these are the phenolic compounds, and it is a major defense, they are used in a major defense mechanism against biotic and abiotic stresses. And because modern wheat varieties, they have very good biotic and abiotic stresses, so that's why they have more ferulic acid contents. So this was the initial result, and we didn't stop there. We did genome-wide association studies. We tried to find out the candidate genes which are involved in the ferulic acid biosynthesis, and we came up with the 13 different loci in the wheat genome which are involved in the ferulic acid biosynthesis. And after the systematic analysis, we narrowed down this region, and we identified that there are actually 3 candidate genes for the ferulic acid biosynthesis, and these genes are actually the novel genes. And if you look up at the expression of these genes, that these genes are highly expressed in the wheat grain at the developing stages. So these expression bars, you can see this is the expression of these 3 candidate genes in the low ferulic acid varieties and in the high ferulic acid varieties. And you can see that these genes are highly expressed in the varieties which have high ferulic acid contents. So again, we try to validate these genes. We want to make sure that we are going towards the right direction, and we have the actual candidate gene which we can use in breeding. So we identified 4 different EMS mutants. These are actually the knockdown mutants, where we have knocked down these genes by the ethyl methyl sulfonate mutagenesis, and we compared the proportion of the ferulic acid contents compared to wild type. You can see that in all these 3 mutants, the ferulic acid contents is almost 12 to 16% lower than the wild type. So it means that we are going towards the right direction. Then we validated the effect that if these mutations are present in the wheat collections, what is the effect of these genes? So you can see that all the varieties which have the favorable alleles of these genes, they have almost 7.2 to 5.6% more ferulic acid content. So now we are doing more functional studies where we can identify that how these genes function in the ferulic acid biosynthesis. So we are doing experiments with the gene editing and by developing the backcross chronos mutants. So the second part of our research is on biofortification. You know, it is very important aspect, and it is related with the human health. So over the years, CIMIT and the Harvest Plus collaboration has resulted in wheat varieties which have high zinc contents. But unfortunately, we don't have any wheat variety which has high iron contents. So that is what we are trying to do, that if we can also increase the iron contents in wheat varieties of Pakistan, because we have very high prevalence of the iron deficiency anemia among the population of Pakistan, especially in the lactating women, in children, and in the elder population. So what we did in the beginning, we tried to identify that what is the mechanism behind the iron homeostasis in the developing wheat grain of a Zincol wheat cultivar. So Zincol wheat cultivar was released in 2016 in Pakistan, and it does not only have the high zinc contents, but it also has high iron contents. So by the systematic analysis of a huge transcriptome by the RNA-seq of the zincol grain, we limited our result to 2 important SNPs which represent the genes which are present in the zincol grain, and these SNPs were actually involved in increasing the iron and zinc contents. So these 2 genes were TA-BZIP14, which is actually a transcription factor, and nicotianamine aminotransferase gene. So in these both genes, the zincol type allele, it can systematically and consistently increase both grain, iron, and zinc contents. So now we know that these genes are actually involved in grain, iron, and zinc biofortification, and we have this knowledge previously in model crops like rice and Arabidopsis. So, but we were trying to look for more novel genes which are present in these wheat varieties, so we can have more candidate genes which we can use in breeding. So what we did next, that we again did the RNA-seq of zincol variety, zincol cultivar, under iron deficiency conditions. So we reduced the content of the irons to the plant. So we stressed the plant to the iron deficiency, and then we identified that what are the genes which are being highly expressed under the iron deficiency. So that can lead us to some of the more candidate genes. So by this analysis under iron deficiency, we identified certain transcription factors, and one of the major findings was that we were able to identify a novel gene which is a alpha beta hydrolase superfamily protein, and this gene consistently increased the grain iron contents in 7 different environments in wheat cultivars. And you can look that the expression of this gene is higher in both embryo and the endosperm. It means that there is good expression of gene in the grains, not only in the leaves and roots. So we identified that if the favorable alleles of this gene is present in the global population, that if there is some opportunity to use this gene in the breeding, so we screened more than 3,000 global wheat collections, and we identified that the favorable gene is in very limited quantity in the land races and cultivars. So this gives us a very good chance that we can use this gene in the breeding, and by using this gene we can increase the grain iron contents of the wheat. So again, the iron homeostasis is a very complex phenomena because it involves the genes which are related to the iron acquisition from the soil, iron transport, and the iron storage. But in all this iron homeostasis network, the most important part is the Brutus or HRZ gene, which is actually a BHLS transcription factor. So it is like the master driver for the iron homeostasis. So what we did, because we have very excellent genomics technologies available, and we have the technology available of the CRISPR-Cas, which is now very efficient technology by which we can knock out any gene. So what we did, that we tried to silence this gene because it is a negative regulator, and we hypothesized that if we knock out this gene, we can increase the iron contents in wheat. So this was actually the work of Dr. Samar, who presented me very well today. So we have the traditional knowledge available about the genes, about this Brutus gene in P and Arabidopsis, that in P and Arabidopsis we have the loss of function mutations of this gene, and by the loss of function mutation there is very high iron accumulation in the leaf and different tissue parts. So what we try to do, that we try to delete this 15 base pair proteins, different 15 base pair segment of the nucleotide by the CRISPR-Cas, and these are the results by the CRISPR-Cas, that when we knock out this gene, we have very good high iron accumulation in both grains and in the leaf samples. So these were very good results. We again try to quantify the iron and grain contents, not only in leaf, but also in the grain samples and white flower. So you can see that all the gene edited lines, they have very high iron and zinc contents as compared to the control variety or wild type. So this was a very good result, and it actually became the part of the UK CGIAR project, which is a joint project between Pakistan, China, Pakistan, UK, Egypt, and Kenya, and we are trying to do the field trials of these gene edited lines in these countries so we can directly deliver the product. So again, because these are the gene-edited lines and there are strict regulations on the commercial utilization of this technology, so we also tried to find out the natural variants of this gene. So this is an extended work, and we identified some land races, some old cultivars where we have the variants of these genes, and we can utilize these variants in the breeding program to increase the iron contents. So this was the second part of the project. The third is translational genomics. I will not go into the detail because I think we have very less time available for the lunch. So actually there are huge advancements in wheat genomics, and if we can count after the sequence of wheat genome in 2018, there are almost 26 very large scale wheat genome sequencing studies. And in all these studies, very large wheat populations, they have been underwent the whole genome sequencing. But how we can translate this knowledge for the wheat breeders so they can use this information in developing tools and resources which can actually help them to develop future wheat varieties. So that is our main work at CIMIT, that we try to translate this genomics knowledge into applied product, where we develop the markers and SNP chips which breeders can use to develop new varieties. So over the years, we have developed several platforms. We have developed the Cas markers, which is a very trademark technology, and almost 40 breeding programs all over the world, they are using our marker platforms. So the showcase platform which I want to introduce today is the Novo 5K chip platform. So this Novo chip platform has 5,000 SNPs. It includes all the functional genes and the QTLs, which we selected from the whole genome sequencing data. And we think that it is an excellent platform for the genomic prediction, marker-assisted selection, and gene tagging. And the cost of this platform is also quite low. So this is the overview of the platform, which include the functional genes, QTLs, and genome-wide SNPs. So we also used this platform to predict some phenotypes. Here you can see a result in a mapping population that when we use this 5K SNP chip platform, we can actually predict very important phenotypes related to yield and important quality traits. So this work is going on. So hopefully by the end of this year, this platform will be publicly available, and wheat breeders across the globe, they will be able to use this platform to develop future wheat varieties. So in conclusion, these new genomics technologies can better help to develop functional and nutritious wheats. Gene editing is a very key technology where we can knock out the master regulators to improve some important phenotypes. So translational genomics, it can actually help us to develop tools and resources for the future wheat breeding, and we can develop platforms to do the large-scale genomic studies for the wheat breeding.
Thank you very much, Dr. Rashid, for your inspiring talk. Your pioneering work in wheat breeding, genomics, biofortification, and functional markers has a global impact, and your leadership will continue to shape the food security and sustainable agriculture worldwide. We deeply appreciate the knowledge and vision you have shared with us today. Now we will move forward to our next keynote speaker, Dr. Benjamin or Dr. Okay. So our next keynote speaker is joining us virtually from Australia. It is my great pleasure to introduce Professor Benjamin Schwesinger, an internationally recognized plant and fungal biologist at the Australian National University. Professor Benjamin leads the group at Australian National University Research School of Biology, where his research focuses on plant-fungal interactions, host pathogen co-evolution, and plant biosecurity. He has pioneered in innovative approaches in genomics, diagnostics, and pathogen surveillance, including air sampling technologies to detect the fungal pathogens. Professor Benjamin also serves as a chief investigator at the ARC Training Center for Future Crop Development. Through his impactful research and leadership, he is advancing global efforts in crop resilience and sustainable agriculture. Please join me in welcoming Professor Benjamin, who is with us online today. Hi, Professor Benjamin. Can you hear me?
Yes, thank you for having me. Great pleasure to be here. Yes, I pre-recorded my talk, so you can listen to it without any interruption, I hope, and I'm happy to answer any questions afterwards. So thank you.
Yeah, sure. So over to you, Professor Benjamin.
Welcome all, and thank you for having me. My name is Professor Benjamin Schlesinger, and I would like to acknowledge the kind invitation by Professor Dr. Sylvia Cranwell. It's great to be here, even if it's just remotely. I've been to Pakistan earlier this year. It's a lovely place to be. I was very grateful to have great hosts at various institutions. So I'm very happy to be back, at least virtually for now. So today I will talk about the weird and wonderful genobiology of plant pathogenic fungi with multiple nuclei. So in most cases, you know, we are diploid, so humans are diploid, so we have two copies, one copy from your mom and one copy from your dad, and you inherit this in one single nucleus. Rust fungi and many other fungi are different, that they have two different nuclei. And I will tell a little bit about what this compatibilization really can do to your genomes. So first of all, thank you to all of the students and staff and collaborators which really enable that work we do. Most of the work I'm presenting today has been performed by a very talented PhD student, Rita Tam. And we also have various collaborators around the globe. So we have collaborators from the Aarhus University in Denmark, very close collaborators. Then we also have two collaborators in Pakistan, Sajid Ali and Abis Rashid, which we really appreciate because the pathogen we work on, they're really global pathogens and not restricted to a single country. So we really have to fight them with a global mindset. So thanks for having me. I think Pakistan is very important for this. So rust fungi. So rust fungi are fungi which are an ancient problem in Western agriculture since we grow wheat. So both the Greeks and the Romans had gods. This is Mars in the Greek mythology, which was responsible for rust and keeping rust at bay. And then in the Roman history, we have Robigalia and Robigus, which was god to appease to get good crops against rusts. So this god was to be prayed to prevent rust infections or whatever it is, like the rusty color stuff. And then Romans normally sacrifice dogs and cats in April, so in springtime, so that they didn't have this thing on the crops, which wasn't really good for them. Of course, I didn't know they were fungus. So what we really work on is rust fungi, the ubiquitous plant pathogen in agriculture and the environment. So we work on stripe rust on wheat. So we also work on myrtle rust, which is a disease from lots of our trees in Australia. And there are many, many other rusts around. This is like an orange rust and blackberries. And we also work on sugarcane rust. So we do have a little bit of history of working with rusts, which affect crops, but also environmental species like trees and shrubs. So rusts are the fungal order with the most plant pathogens described, so roughly 8,000. The obligate biotrophs means they can only grow on the plant and can't be cultured on plate. They're very important for agriculture because they really infect major crops. So if you had a piece of bread today, you kind of competed with that pathogen for their food source, and we kind of won this time. But other times the pathogen wins and devastates fields, and that's what we want to keep at bay. There's also coffee, soybean, corn, switchgrass, barley, sugarcane. Many plant species are infected by rusts, and rice is one of the exceptions. And they are important for ecosystems too. For example, in Australia, myrtle rust is very important. But today we're really going to focus on stripe rust. So stripe rust is an important pathogen of wheat in Australia. It leaves these stripy infections on wheat. It produces these spores which are dikaryotic, which have two nuclei, and then can reinfect again the same wheat crop and have really this amplification of infections. So University of Sydney does the national surveys, and you see in 2022, for example, we had many striped rust infections on the East Coast, so it's a major pathogen in the East, but there's not much in the West, even though we grow a lot of like wheat here, basically here, and then here is our wheat belt. That's where we grow wheat. So we see the pathogen in the East, but not really in the West. So as I started the talk, you might be familiar that you have diploid organisms, and the weird and wonderful world of fungi enables these eukaryotic organisms, or actually in some fungi also many nuclei, from tens to hundreds to maybe thousands even, is some of the mycorrhiza. These fungi we work on at the rusts, in the dikaryotic stage, they have 2 nuclei, and this is a stage where they infect most of the crops and really lead to epidemics, because these spores in the asexual cycle. So we have this nuclear compartmentalization, and so what our goal is to reconstruct genomes where you can say one genome copy belongs to A, another genome copy belongs to B, and so what's the nuclear complement of the genomes. For striped rust, we have 18 chromosomes, so 2 times 18, so 18 haploids, so 36 total. Each nucleus has 18 chromosomes. We have 50% repeats. A little bit more background on the rust fungi, why they maybe have been very well studied on one side, but molecularly and genomically, we just they're really only now reaching maturity to fight them. So these striped rust fungus, they have like a very complex life cycle. So in the western wheat growing areas, we have mostly only have the asexual stage, but in Pakistan, in the Himalayan region, in Nepal and China, some of it of India too, we do have the complete asexual cycle where we have karyogamy, recombination going on, and then sexual populations. But this seem to be for striped rust only the Himalayan region. That's why it's so important to work with you in Pakistan to study this fungus, and we hope we can extend our collaboration in the future. It's typical to complete the life cycle in the labs. There's only 2 labs reported, one in Denmark and one in China, which can complete the full life cycle. So it makes it difficult to cross between isolates. They're obligate biotrophs, they're only cultural on the plant. They're difficult to purify because you have to get these single pustules, so single lesion on the leaf and then purify it. It's not like you can grow on a plate where you can pick a single colony. It doesn't work like that. They are dikaryotic in the epidemic stage, so they have 2 nuclei, which makes it exciting, but also makes it more complicated. They can have very complex genomes, very large. The largest are right 3 gigabases. Lower ones, like striped rust, is thankfully smaller because we have haploid genome size of approximately 80 megabases. And there's no heterologous expression system for wheat and for many other hosts, so we can't study effectors really well, I would say. There's some publication in China which do this better than we do. And then there's no stable transformation system, so we can know gene knockouts, knock-ins, so it's really hard to study those. But we have overcome some of those challenges most recently, so we have generated heterologous expression systems, and we really tackled the genomes. Today I'm also only going to talk about our genome work and not about anything else. So, talking about genomes, I give you a little bit of an introduction of where genomes came from in terms of genome assembly over the last decade, last several years. So coming back to this dikaryotic nature, we would like to know what the nuclear complement is, so what the chromosomes within one nucleus versus the chromosome with another nucleus. So we really would like to have 2 sets of 80 chromosomes, and we can say this set goes to the one nucleus, the other set goes to the other nucleus. So where do we start? Oops, cut off here, apologies. So really, when I started to come in here in the field was 2015, and basically what people could do is assemble with short reads, Illumina sequencing, and then what I call the cut up spaghettis, so or noodles or soup, like basically cut up your strings of chromosomes and then you don't know which piece belongs to which, and you can't know which piece belongs to which nucleus either. So they were kind of good for gene content, maybe, but really there was lots of issue with those assemblies. They were good initially at the time, but really I would not reuse them now because there's no haplotype information, there's no genome structure, they're highly fragmented, there are lots and lots of errors. So when I came into this field, so it was with noisy long read, so PacBio sequencing till 2019 and nanopore sequence until more recently. So what we kind of could get is these longer contexts. So from 1,000 contexts, we go to 100 contexts. We have some phasing information, but we still have phase switches, and we have some information. We don't know which pieces belong together, so we have no nuclear complements most of the time. But I contributed at the time to several publications, which is here in purple, which used some of those technology. So they were good for gene content, they're good for resolution of transposable elements, starting points, and initial insight into haplotype variation. So it's exciting times really to start genome biology fungi. Oh, an initial insight into genome structure. Sorry, apologies, it overlapped. I copied this from a different computer. So we do a little bit of genome structure as well. Oh, and start for proper Rust genome biology. I really apologize. Different versions of the same program on different computers. Apologies. So really, there was a time where we could really look into genome biology some more, but we weren't really there yet. Because we did have residual errors. We still had errors in here. So what really enables us to go the next step, or even to this step already, is the free sharing of protocols. So we shared many of our protocols on Protocols.io before we published them, and also shared videos of how we extracted DNA, because extracting DNA is very hard from fungi, especially if it's really long DNA, to get the sequencing done, and many people share the pain, but we are really happy to help if anybody had problems with these. And then the other technology, which was very important for Rust fungi, was this Hi-C. So this is a technology which captures the 3D conformation of DNA in a nucleus. So it captures what pieces of DNA interact with each other. And because the Rusts have two different nuclei, it was very useful to say which piece of DNA comes from one nucleus and which piece of DNA comes from another nucleus. That was very, very useful to have. So what could we kind of gain from this initial investigation? So this was the first paper with really long read genomes, not perfect, but what you could show is that the variation between the two genomes, the two nuclei, partial pseudohaplotypes, is really mostly in large variations. So variation between 500 base pairs to 10,000 base pairs. So most of the variation is actually not in SNPs. SNPs is only a small part of it, like these very short, small deletions, insertions. Really, most of the variation between nuclei is in large chunks of DNA, but we nowadays call structural variations. But we could also contribute to like early work on that Rust fungi can exchange complete nuclei to evolve and generate more virulent isolates. So that was shown for a standard isolate, where we contributed some of the analysis. Most importantly, I myself actually made some of these Hi-C libraries to be able to phase these genomes out into nuclei, which was essential to release these discoveries. So this was with collaborators at the time at CSIRB. And then some more work from CSIRB, which we contributed to at AIOU, is to really make complete chromosomes out of this and to scaffolding, look at some genome biology and methylation, centromeres, and so on. So where are we now? Right now, we're really at complete genomes because we have high quality long read sequencing. So we just recently published this actually in genome biology. So we can look at telomeres, centromeres, and RNA repeats. We can look for haplotype specific expression and transposable elements and also genes. I'll show you a little bit of this. And really, there's minimal residual error. And really, we are ready for large scale whole genome population studies, which I will show you today. So the initial finding, again, from the technology was that high feedback bio was better than noisy nanopore data at the time for phasing. That was work we contributed to as well, led from CIRO. And then CIRO also went on their own and studied some beat leaf-rest isolates and hypothesized certain recombination events as well to traumatic exchange. I think they hypothesized. They showed some of them, they hypothesized others. And then what we showed is really in mating types. So if you look at fungi from our lab, we looked at certain loci in fungi which control mating, but also probably nuclear pairing. And so we studied them in quite extensive details as well and showed some about synteny and genobiology of those as well, which we published last year in PLAS Genetics. So really, again, striped breast had an asexual cycle and a sexual cycle. So we're working with Aviz and also Sashid to get isolates from the sexual cycle, like DNA from isolates and more from them. But really, right now, let's focus on the asexual stage. It's the absence of the sexual cycle. So let's look what happened in the Western wheat growing regions. So an example in Australia. So in Australia, we have the first incursion. So we don't have any asexual cycles, only like asexual propagation. So we have the first incursion 1979, which is here. So we have only one lineage. Then we have new incursion around 2002, which then took over very rapidly. So replaced the previous isolates. And then in 2017, 18, and 20, we do have several other incursions into Australia, which kind of really changed the picture because now they kind of compete in the landscape. This is really work from University of Sydney, but I think it's very impactful because it's important to have this long-term surveillance data across the country with quite comprehensive data to see what's going on. So let's first look at one isolate first to get a little bit of an idea how this genome of an individual isolate looks like. So we focus on PSD S0, which we have been studying for quite a while. So if you do this genome assembly from PSD S0, which is was done by Rita Tam, which was published this year in Genome Research, is that we could reconstruct 18 chromosomes. Most of them were complete. We have one single telomere missing, and all these other telomeres we could either find in the assembly or reconstruct. And we have some smaller gaps, which are mostly around the mating type loci or the RDNA array. And so how does it look like in the signal? If you look at nuclear compartmentalization, this is an interactive heat map, interaction heat map of the high-C data. So you see that we have one set of chromosomes interacting quite well with each other in one nucleus, and another set of chromosomes to interact in the other nucleus, which is A and B. And this is kind of the genome compartmentalization, which we can really see nicely in the data that these only interact with chromosomes which are in the same nucleus. So we did lots of different studies in the paper. I just give you one brief highlight. Now we have completely phased genomes, and we can look at allele-specific expression. So if one allele is expressed more than the other. And so that is what we find. So in roughly 7% of all genes, we do see that we have allele-specific expression when we analyze an expression time course, which goes from uninfected, from spores to several infection stages. And we can also show that these are enriched in affected genes, the genes which are important for the infection process. So up to 15% of all, 15% of the one-to-one heterozygous genes display allele-specific expression, and this is really enriched for effectors and secretory genes. Then we could actually also look at the methylation status, because our nanopore data also gives the methylation status. So we use nanopore data for assembly. And we could basically show that in differentially expressed genes, that the allele which is lower expressed, the green ones, has a higher level of gene body methylation than alleles which have no allele specific expression. The low allele specific expression, both of them are very lowly methylated across the gene body, but if you have allele specific expression, then the allele which is lower expressed has more methylation, which is kind of cool. So we also did lots of other studies. I would really encourage you to look at the paper if you're interested in this some more. And we can also show that it's really enriched for secretome genes, which are allele specific for expression. So from one genome, we went into multiple genomes. So now we have assembled many more genomes, most of them with us, and planned by Sydney University also contributed to this, and Denmark also contributed to this. And then we also use public data. So really, what's the genetic diversity between lineages, which I will tell you now. So now we have the complete set of nuclear phase genomes. So we can now do analysis on individual haplotypes, on individual nuclei. And so this is a PCA, like a principal component analysis of the genetic variation which we see between nuclei, not isolates nuclei. And we see that we have two groups of nuclei. So we have an A group and we have a B group in the western regions, or the one with this one Chinese isolate. And since it's A nucleus and a B nucleus, and they're really quite distinct because it's 60% of the variation. And we have more genetic variation in the B group than in the A group. So that was quite surprising, because normally you would expect much more in between thing. So what did we do? So and then we do a different approach, not a PCA, but actually a principal component analysis, not a principal component, phylogenetic approach too. We get this A group and the B group. We see that we have quite good synteny even between the A and the B group. The chromosomes do seem to be conserved, but really we do have some inversions and duplications. Most of those are really related to the mating type locus, which I spoke about briefly before, and the other one is the rDNA array as well. So we don't have like any major large rearrangements except for these two different loci. So what do we do if we actually want to compare all the genomes with each other? So we have to put them in a graph. So we have to put them back into a graph. So genome assembly basically builds a graph, distangles the graph to get chromosomes, and now we have all these chromosomes, we mesh them all together. So what we basically do here, what is a genome graph? So if you have two different haplotypes, so they're in this node, they share the same sequence. In this bubble, they have distinct sequences. Then there's a node again, and then where you do have a deletion, for example, you do have a bubble too, where you only have an edge for the one path, but a node in the other path. And then SNPs just all the bubbles with just one base pair long. And then, let me move this so you can see. And then what we do a pan-genome variation graph. So we do have bubbles where these two haplotypes are different, or maybe also a repeat and so on. And then we put it all back together, and then we can investigate the graph. So this reduces reference biases, maximizes the capture of genetic diversity, and reveals complex regions. So then we can ask the same question, what haplotypes are related to each other, by looking what shared paths they have. So how similar these different paths are for different nuclei. And that gives us a very similar result. So we do have a set of nuclei which share very similar paths. which is the A group, and then we have more nuclei in the B group, which more are like the path. And this is a really nice analysis because compared to k-mers, you do actually have the haplotype information contained in the graph, while k-mers are really just individual data points where you don't have any haplotype information about them. So this graph-based analysis is quite neat. Analysis is very similar to the phylogenetic analysis, which is very similar result to the PCA, to the principal component analysis of diversity. That we have two major groups which we currently capture, A and B, in terms of nuclei. And then also, if you do a network analysis, again, you have an A group and a B group, and the B group is more diverse, with more reticulation than the A group. Of course, the question is, do we have something else? Because now we have only A, we have only B seen, but it's the best, right? And we might be blind to other things. So then it became really important to collaborate, and so Kava Majid, he was visiting PhD student with us. I still have to write a letter for you. Apologies for being late on this. And then we did a phylogenetic analysis of these samples Kava brought to us after quality control and all that stuff to really look how these Pakistani samples look like compared to anything else. And we see that the Pakistani samples are really quite different to what we see in the West and in the phylogenetic tree. And then we also wanted to go further in k-mer analysis because we don't have long reads. So right now we can't do the path analysis in the graph, which we will get to next. But now it's k-mers, just the general base pair composition of these isolates. And if they kind of isolates could have more diversity, different nuclei, different genetic components than we see in A and B. That's what we can look at with these k-mers. And what we can see here, in Pakistan, you have many isolates which are basically potentially AB. So basically here, all of those, they're basically AB. This as a reference is a BB isolate. So we have lots of k-mers which map to B, and not as many k-mers which map to A nucleus here. These are all ABs, though they both light up in the B nucleus and the A nucleus. But then we also have some other very interesting isolates which we now analyze with our graph approach too. Is we do have A isolates and maybe also AC isolates, so like a completely new nucleus as well. So that's quite interesting. That's why it's important to work collaborate internationally, and I hope we can extend this. So these are some of those. So we have some AA isolates and AB isolates, and potentially some isolates which are potentially AC as well in here. And so we're analyzing them against the graph because we want to know if these regions, how certain regions look like in the graph when we map these AA or maybe AC isolates back to the graph. And maybe find new ways in the graph which we have not seen before. What do we know? Just to wrap it up, not for me to go over too long, is that we also use this data for nuclear exchange in striped rest. So we worked with people at the Global Rest References Center quite extensively. So they used our data set to place molecular markers, SSRs, on these chromosomes, which is very useful. And this then allows them to postulate certain hypotheses around somatic hybridization. And we then confirmed this with whole genome sequencing. So we could show that in PSTS0 isolate, in a PSTS7 isolate, each had donated one nucleus to an isolate, which gave rise to PSTS10. And then we could show this many more from the extended data sets which we have not published yet. So somatic hybridization, nuclear exchange seems to be very important outside of the Himalayan region for striped rest to reassort nuclear complements to give rise to hypervirulent isolates, because all of those hybrids rose to very high frequency in the global population in the West, which is mostly focused on the West. So that's quite interesting that somatic hybridization contributes to variant evolution, and we really need to look at whole genomes and nuclear genomes to be able to hypothesize and then also confirm this. What are we doing next as well? So we look at graph genome and analysis based on variations, which we see in short-read data set mapped against the graph, and I have a couple of questions about genome evolution in this, especially with a focus on Pakistani regions too. We're also interested in DNA methylation because we know we see DNA methylation that contributes to allele-specific expression. We potentially also will look at, if we get some money, to histone marks as well. And then maybe one day we look at RNA modification if we think that's important. But we definitely already have some data for DNA methylation between isolates and overlays this with expression changes as well. So just a highlight of this, so we have 5-MC methylation in a Danish isolate versus an isolate from Australia, and you see that there's much more methylation in this gene region in the Danish isolate versus the Australian isolate, and we think that this DNA methylation contributes to expression differences between isolates, not only within an isolate, also between isolates, which also can contribute to virulent evolution beyond genetic changes in the pathogen. That's all for now. I hope you enjoyed the talk. I have to cut it out a little bit together. Hopefully it already happened. And I'm happy to answer any questions and apologize if some of the slides got a little bit mixed up. It was a great pleasure. And yeah, happy to answer any questions. And I'm live online. Thank you. Bye.